Here we go. All right, so I know we're past this. We were further, right? Oh, we weren't. This way?
We were at their cell cycle class.
Okay.
Yeah.
All right. Here we go. All right. So we are looking at then, starting to look at two separate molecules that are involved in controlling cell division or controlling the cell cycle. And those two molecules are cyclins and cyclin-dependent kinases. And I believe we reviewed what a kinase is, right? That it's a molecule that enzyme that phosphorylates molecules and activates them. And so these cyclins and CDKs, which is short for cyclin-dependent kinases, fluctuate and go up and down, and it's the concentration of these that is helpful in triggering the cell to go through the various stages of the cell cycle. So in looking at this graph, I didn't talk about this really, did I? Did I talk about this? No. So on the x-axis we have the time, and if you look up here, one of these arrows represents one cell cycle. So the cell cycle has G1, S, and G2. Those three comprise interphase. And then they enter the M phase, which is mitosis and cytokinesis. So the cell will divide into two. Then those two cells enter in their own cell cycle. So then they enter in G1, S, G2, and then will divide, and so on and so forth. So what we're looking here is, at the beginning here is, let's look at the cyclin concentration here. The cyclin concentration, as we just wrote, fluctuates, and we see that with these peaks and valleys here. So let's look at when it peaks. So the cyclin concentration this is the end of the G2 phase here, so it's going up, 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 until it gets into the M phase and then drops back down and stays constant for a while until the next S phase, where it begins to increase in concentration into the M phase and drops down, and so on and so forth, and follows that pattern. Now this MPF activity, MPF is an activated CDK, basically. And so for right now, we'll talk in a minute about what MPF is, but what this does, the activity seems to follow the increase of cyclin. If you look here, the cyclin increases, and soon afterwards, MPF activity increases. Cyclin decreases, MPF activity decreases. And that follows that same pattern. Whenever cyclin increases, then the MPF activity increases. And so this molecule cyclin seems to control the activity of this molecule called MPF. So let's look at why that is. So in this picture here, it shows you the cell cycle. So here's the G1, S, and G2 phase, and then the M phase. So let's start here with the beginning of the cell cycle, and with the CDK here. I'm going to put here that this is an inactive CDK. So it's present in the cell, but it's not doing its job yet. It's not in the right shape. And this CDK is present through all of interphase. So here it's present, and here it's still in the same shape. It's still in the inactive form into the G2 phase. But what happens starting in the S phase is this other molecule, cyclin. Remember this CDK stands for cyclin-dependent kinase. So here in the cell, we didn't have cyclin, and so this is inactive. It's an inactive CDK. Now what happens is in the S phase, this arrow here, notice it starts at a point and gets thicker. That's illustrating the accumulation of cyclin. So in the S phase, the cyclin begins to be made in the cell, and so it starts to accumulate. So we have an increased cyclin concentration in the cell at this particular point. So why does that matter? So cyclin, what does it appear to do, according to this picture? Yeah, this is what activates the CDK, and when it's activated, when the it activates it by binding to it. So they try to show you here the shape, complementary shapes here. So the MPF is really the cyclin bound to the CDK, and that's your MPF. This is basically an active CDK. All right, it's activated the CDK. So cyclin, we can write here, binds to the CDK to form MPF. And MPF, I'm going to put in parentheses here, stands for maturation promoting factor. So MPF. But what it actually does is once the cyclin accumulates and creates this MPF, that's the molecule that triggers the cell to get past the G2 checkpoint and go into the M phase and divide. And so another way to say this, instead of maturation promoting factor, although that's the technical name in the MPF, we can sometimes the textbooks will say you can think of it as mitosis promoting factor, because it's promoting the advancement from interphase into mitosis. They both happen to start with an M. All right, so it works. So MPF, we said, is an active CDK. CDK is the kinase, which, what do we say? It phosphorylates molecules. So I'm going to write here that the MPF, let's write here, that it phosphorylates proteins that promote mitosis. And so, as we enter, now it goes into mitosis and cytokinesis, where the cell will eventually divide. And so the MPF has done its job, and now we have cyclin is degraded. These little cracks here are supposed to represent the cyclin being broken down or degraded. So your degraded cyclin goes off, and the CDK goes back to its original shape, which is in the inactive state, and it'll stay inactive until the next cell cycle where the cyclin accumulates, and the thing will go around and around and around. And so cyclin, a very, very important molecule, and so the presence of cyclin is important in getting into the M phase. So if a cell didn't have cyclin, what wouldn't happen? Yeah, mitosis wouldn't happen. It wouldn't make the MPF, it wouldn't activate the CDK, so the right proteins wouldn't be phosphorylated, and so on. So going back to and relating this to this picture here, so this graph, so let's erase this part here. So during the S phase, cyclin activity or cyclin concentration goes up. That's right here. S phase, cyclin concentration is accumulating to the G2 phase, and then it binds with the CDK to make MPF, and the MPF becomes active. Whoops, wrong way. So therefore, kind of here at the beginning of the M phase, MPF becomes active. Cyclin then is degraded, goes down, and MPF then also goes down. And so therefore, cyclin degraded, MPF goes down. All right, so that's how those things work. And so that's one mechanism of controlling the cell cycle. Why is it important for us to control the cell cycle? Why it's important?
Well, we need to control the cell cycle because if there's like a mishap in one of the sections, then it will just go on to mitosis
absolutely. Shane?
Okay. So if a cell, like let's say there was something like a mutation in the DNA that did not allow the cell to create a cyclin, would the cell just go through the G1S, G2, and then back into the G1 and just get so big that it was going to burst?
No, it would just stay. It would never get past here, so it would stay here.
It
You
just
know what I
wouldn't
mean?
divide, it
Yes.
just-
Yeah. So I mean, if that happened like in the very early stages of development, there would be no organism
Right.
that's made, so you wouldn't be able to divide the cells. Absolutely. All right. So so let's talk about other. So that was an internal signal that the presence of cyclin is an internal signal that helps to control the cell cycle. We're going to talk about some external signals. So another internal signal that we've talked about is with the checkpoint. So an example of an internal signal is that kinetochore is not attached to the spindle. Microtubules send a molecular signal that delays anaphase until the kinetochores are the spindle fibers, the microtubules that attach to the chromosomes to separate the chromosomes to opposite sides of the cell. And so that's part of the M phase checkpoint, and some of those that won't go beyond that. So all the checkpoints, the cyclin and the CDK are all internal signals and it's going on inside the cell to regulate and control the cell cycle. But there are some things that are external outside of the cell that can also control the cell cycle as well. They have some influence on there. So an example of that is a growth factor. Growth factors are signal molecules. They're like the ligands. They're a type of ligand that we talked about in chapter 11 with cell communication. So these are proteins released by certain cells that stimulate other cells to divide. And so I'm going to show you an example of one with this as this platelet-derived growth factor, or PDGF. This stimulates the cell division and growth of human fibroblast cells. Fibroblast cells are a certain type of connective tissue in humans. And so, and it says platelet-derived. Do you guys know what platelets are? Yeah, they're fragments of blood cells that are in your blood that they're involved in clotting of blood and so on. So these are cells that are in your blood, or pieces of cells or parts of cells that are in your blood that secrete this growth factor and will stimulate the connective tissue to divide. So, so what I want you to do is, we're going to look at this. Actually, I'm going to put this up on the, because I have one thing I want you to write on this. I guess I can just read. All right, so when you're looking at this, mine's color-coded so you can kind of see how they match. So this one right here says without PDGF. So what they've done is taken, these are human fibroblast cells. So these are human fibroblast cells. They're cutting them up and then they agitate them, and so you have a suspension of cells. And then, just as if when we talked about growing bacteria cells and the agar on a petri dish, you can do this with human cells as well. So they have the agar here. So this contains nutrients and so on for the cells. And the cells can grow on this. And so I don't know how well it copied on yours, but there are cells here, all right, on there. So this is without, and remember this is the growth factor. And then they took the same types of cells, the same human fibroblast cells, and plated them in a culture with PDGF. And so this also contains nutrients. So this is the same agar, except this one has the growth factor and the first one doesn't. So when we look at and compare these, so what do they color code this? So this without the PGF, this is the before. So they plated it. This is what they look like at the beginning of the experiment. And see, mine is color coded. This is after. So these two go together, these two pictures here. All right, before and after of the cells without the PDGF. And then with the PDGF, this is the before, and this is the after. So these two pictures go together. And does the PDGF seem to make a difference? Absolutely. So what they found is that this growth factor, when it's not there, even though there's plenty of space, they're giving the cells nutrients and so on, without this growth factor, these fibroblast cells do not divide. But given the growth factor, they obviously divide a lot because you have a bunch of the cells in here. And so the growth factor, what it does is if this is one of the human fibroblast cells, you have receptor proteins. It's actually a tyrosine kinase receptor they found is one of the receptors. And the growth factor, so this is the PDGF, binds to that receptor protein. That triggers a signal transduction pathway that eventually makes molecules that cause cell division. And so those molecules aren't activated that contribute to it entering the cell cycle unless PDGF is present. So we can write here the binding of PDGF to, they're actually tyrosine kinase receptors. The receptors in the membrane triggers a signal transduction pathway. Resulting in cell division. Which explains why the cells without the PDGF are never divided. Signal. So something outside of the cell, there's not any the molecules inside the cell aren't controlling it. It's an external signal. So growth factors, with this being one example of that. A couple of other examples of external signals is something called density-dependent inhibition. What that basically means is when cells get crowded, they stop dividing. That's just what normal cells do. Another thing that normal cells do that has to do with outside the cell is they exhibit what we call Anchorage Dependence, meaning that says that which they must be attached to a substratum. That's a surface. They have to, so it could be another cell, a layer of cells. It could be, agar, a petri dish. They have to have a surface in order to divide. Normal cells need these, so this is part of the external environment controlling whether or not the cell is going to divide or not. So this picture here shows this. So along the top here, you have cells that are anchoring to a dish. That's the Anchorage Dependence, which will allow it to divide. So if they don't have that, our cells will not divide. And then notice here that there's space. So what happens is these cells will begin to divide and take up that space. But notice here that once they get into a single layer and there's no more space, that's the density part, then cell division is inhibited. So that's why the inhibition is dependent upon the density. So that's why it's called density-dependent inhibition. And the reason why that is is that when adjacent cells come together, that receptor proteins, I just used the example of a receptor protein binding with a signal molecule, but they also can bind with, I'll put a little shape here, they can bind with proteins in adjacent cells. So here we have two cells that are adjacent to one another. They have proteins embedded in the membrane. When they bond together, so this one here, when it binds to this and this one binds to this is like it binding kind of to a ligand, and it can trigger a signal transduction pathway that leads to making molecules that will inhibit cell division. So they stop cell division. And so then what happens is in doing experiments where they've scraped out or given the cells room now, so now these cells surrounding it right here, these cells right here, do not have this going on, and they will start to divide and take up that space until there's no more space, and so then they stop dividing. And that's how normal cells behave. So they have to be attached to something, and they stop dividing when it gets crowded. So we're going to look at what happens next when these rules or these things in place to inhibit the cell cycle or control the cell cycle aren't abided by. And so basically the rest of what we're going to talk about is how mitosis and these things that control the cell cycle relates to cancer cells. Okay? So, question?
Do airborne viral infections still need a substratum, or do they just not do anything until they get to the host?
Yeah, the viruses, they have to, yes, they don't do anything. They have to inject their genetic material into the host cell and use the host cell. All right, yeah. So the picture below, the picture we just looked at, shows what happens with cancer cells. And cancer cells don't abide by either the density-dependent inhibition or Anchorage dependence as well. And so you can see here doing the same kind of experiment with cancer cells. Normal human cells would make that one layer and stop dividing. Cancer cells will not. And so therefore they will continue to divide, and so you get this like piling up of them. And when this happens in the body, we would call this a tumor. All right, yes?
Isn't that what skin cells do normally though, because they stack on top of each other?
Yeah, there are certain, it's not though, because when they stack up on top of each other, the stratified, we do have certain stratified, we have squamous, like different stratified cells, so that's normal for them. You're right. But not all cells, that's the case. Okay.
Isn't it with like cancer cells, they don't know when to stop, like skin cells when they do that, and that's when they have like a...
Right. There's a set number, like there's programmed in, yes, yeah, that's right. Cancer cells will, as long as there's space and they have enough nutrients and things like that, they will continue to divide. So there's not stopping to them. Yes.
So how would a malignant tumor go from malignant to benign? From growing nonstop to present stopping. So how would it stop?
Okay, keep that question for a second when I'm going to talk about malignant and benign tumors in just a second, then we'll come back to that.
So, when someone has cancer, when they take medication, does the medication like target the surface proteins to like stop
Different medications like chemotherapy drugs will do different things to stop various points of the cell cycle. So like some chemotherapy drugs will, like it freezes the spindles. So basically they can't go from metaphase to anaphase and finish cell division. So different drugs affect and target different parts of the cell cycle, basically trying to stop the cell cycle from happening. And so it depends, different ones work a little bit differently. The one I was talking about is a very common one called Taxol that does that. And so on, the thing is with that though, is that those drugs will also target other actively dividing cells in our body as well. And so that's why with some chemotherapy drugs, the hair loss takes place because the same drug that is stopping the division of the cancer cells or halting or slowing down the division of the cancer cells is doing the same thing to your hair follicles. And so, so they don't have the division there anymore. And so some of the side effects are because of that, like oftentimes after chemotherapy you have a suppressed immune system because they affect some of the cells of your immune system. Nausea, some of them affect the intestinal cells and so on. And so you get some side effects there as well. All right, absolutely. All right, and so they don't exhibit density-dependent inhibition or this Anchorage dependence, meaning that cancer cells can break away from the tumor and still be able to divide. So let's talk about this. Cancer cells do not respond normally to the body's control mechanism. So they're not responding normally to the checkpoints. They're not responding normally to the density-dependent inhibition, Anchorage dependence. And so they form these tumors or masses of abnormal cells within otherwise normal tissues.
Would a cyst be considered like, I mean, not cancer, but?
It's not cancerous, no. So no. If abnormal cells remain at the original site, we call the lump a benign tumor. And these are considered non-cancerous. If they stay at the site, they don't seem to be impairing the tissue that they're part of. They're not inhibiting that tissue at all. In terms of impairing the function, it's called a benign tumor. Heard of that word before? So everybody's heard of it, if you were listening to Joe's question. Malignant tumors, a benign tumor can become a malignant tumor. Malignant tumors invade surrounding tissues and actually can impair their function and can eventually metastasize, which means that they can kind of break off their exported cancer cells to other parts of the body and can form secondary tumors there. And so this picture here shows the example with breast cancer, which can be found in males and females, breast cancer, all right? A lot of people only think of it as females, but males can get breast cancer, it's just more common in females because there's more breast tissue, all right, in females. And so we have at the beginning here a single, notice here it says a tumor grows from a single cancer cell. So you have one abnormal cell that something has gone wrong with it in terms of it not adhering and abiding by the normal cell cycle controls. So therefore it divides if something's wrong with it, and it doesn't stop dividing, so it continually can divide. And so you get this mass of these abnormal cells, and they're all abnormal because it started out with one abnormal cell. Remember mitosis, you get two cells at the end that are genetically identical, and so now you have two abnormal cells, and then they divide, and then you have four abnormal cells, and so on. And so if it's in the tissue and it's not impairing the function of the tissue, it's a benign tumor. A benign tumor can continue to divide and invade the tissue and become a malignant tumor. And so, when you get to this point here, the malignant tumors, cancer cells have also been found to not only bypass the checkpoints of the cell cycle control and so on, but part of what they've been found is that they actually make their own growth factors. So if I'm a cancer cell, they secrete their own growth factors that then can bind to their own proteins on their surface and trigger them to divide. They can trigger other cells to divide. They've also been found to release signal molecules that promotes capillary growth. Capillaries are blood vessels. So the blood vessels, your blood vessels, you actually divide and make new blood vessels that go into the tumor. Why is that important for the tumor? Food and oxygen and nutrients, that's absolutely. And so, it stimulates this growth of the blood vessels in there. And so then what happens is, remember that cancer cells don't adhere to the Anchorage Dependence thing. And so then the cancer cells can break off. You can have a cancer cell that detaches itself from the tumor and can diffuse into either the bloodstream or the lymphatic system. And both of those systems have those vessels, remember, that go all throughout the body. So this cancerous cell is moving through your bloodstream, and at any given time, it can leave the bloodstream, leave through your capillaries, and adhere to some other part of your body. So therefore, a tumor that starts on your stomach, if left undetected, and that cell breaks off and goes into your bloodstream, it can end up, that cancerous cell can end up coming out and attaching to your pancreas, and then can divide and then have a tumor on your pancreas, and that's a secondary tumor. And in layman's terms, that what people say, that when the cancer has spread, that's what they talk about, all right? And so, so that process of the cell breaking off and moving to another part of the body is called metastasis. And I said that it has metastasized, all right? And that just means moved to another part of the body as a result of that. All right, and so when finding a tumor, obviously it's best to get it in the early stages. So like can remove the tumor, and the earliest stages, the better chance that it hasn't invaded the tissue or metastasized yet. And so when they take the tumor out, they usually take some normal tissue around it, they test the edges of that, making sure all the cells around where they took it off are normal. Because you want to make sure you get all these abnormal cells, because if you leave one abnormal cell behind, that abnormal cell can begin to grow and divide, that sometimes people say the cancer came back, all right, and so on, and that can be the case there, all right. So back to your question then. Your question was about
how
so
a
working
malignant tumor can
backwards.
go to benign and vice versa.
So you're saying a malignant tumor after therapy, like?
No, just like as a tumor, like it starts growing, right?
Right.
And it, like as a malignant tumor, why would it stop growing? Or as a benign tumor, why would it become malignant? Are there like triggers or?
That's a good question, and there's in every type of. It depends upon what we're going to see in just a minute. That ultimately what causes this uncontrolled cell division is. Does anybody know what ultimately causes it? DNA mutation. So what does that mean? If you remember that your DNA is double-stranded, and the backbones are phosphate sugars, and you have the bases, A's, T's, C's, and G's. You remember this coming off of it? And so you have these order of bases that make proteins, and these proteins, it's the order of these bases that make these proteins that help to control cell division. And so what happens is, in a mutation, it's the order of these bases that gets changed. And if you change the order of the bases, it can change the protein that's made. And so depending upon what type of mutation, I guess, because there's lots of different ways that the DNA can be mutated that can affect the cell cycle. So I'm not sure exactly, but it could be whether or not it has enough nutrients, enough blood vessels. There's lots of different things that could play a role in that. Like I said, some cancer cells make their own growth factors, some do not. So it just depends upon what the mutation is. So, right, it's kind of a, specific.
Yeah. Is there a difference between regular tissue cells and like cancer cells, like structurally speaking?
Yeah, some of the cancer cells have been shown to actually, some of the mutations can change the receptor proteins. Remember when we talked about the immune system with recognizing self versus not self? And so some of them have been shown to can change the receptor proteins to not adhere to apoptosis and not be triggered, be killed off, all right, when it's supposed to be killed off by signal molecules and things like that. So in terms of that, but that's what they're looking for. There are some, I think, depending upon the cancer cell, like the type of cancer, I think that there are some structural differences that you can tell, like underneath the microscope. Quite honestly, I don't know for what those are for various, you know, different diseases, different types of cancer. Alrighty. So underneath this, you have some room. I'm going to have you write a few things. So I want to talk a little bit about some causes of cancer. So we already used the word mutation. But this process of transformation. Transformation is the process of a normal cell being converted into a cancerous cell. So we said a cell becoming cancerous and therefore not adhering to the cell cycle ultimately is can be caused by mutations in your DNA, which changes the production of your proteins, which then if the mutations are in genes that are involved in making proteins that regulate the cell cycle, then we have a problem. My next question is, what causes those mutations? Like, how does a cell get a mutation that might ultimately cause it not to adhere to the regulations of the cell cycle? How does a cell, what kinds of things could cause a mutation to happen?
Like an error in DNA replication?
Yeah, so normally our cells replicate their DNA when they divide, and we have some mechanisms in place to correct it if we make a mistake in replication, but sometimes we can't correct it, so that can absolutely be a cause of mutation.
Could it be when it goes into the G0 phase and for some reason apoptosis isn't triggered, it could start dividing?
All right, but that's not causing a mutation to happen.
Okay.
So, like, what would trigger the DNA to actually change
Okay.
order of bases that would change the proteins being made?
Isn't there like proteins between the DNAs? Like, isn't there? So like maybe it's like one of the proteins, different protein that acted somewhere as what it should be went in, but like doesn't act the same. It's like,
So protein went in.
so like okay, so like it went in place of another protein, but it was similar, but what it does is different.
All right. Yeah. I like your thought. How would that change the order of bases in the DNA? Because the proteins are kind of like the spools that the DNA wraps around.
Could
So
it be like an R group changing, like attaching to the DNA, and then like maybe altering the code?
I guess it could. That could be. That's not the. I like where you're going there, but it's not one of the main causes of mutations.
What is like a part of the DNA didn't replicate and was missing?
Okay, so part of the DNA was actually missing.
Yeah.
All right, so that's the mutation is missing part of the.
Like if we like missing like the gene for stopping when this cell around.
Okay, all right, so you're totally missing the gene, absolutely. So then you wouldn't make the protein at all. All right, absolutely.
The cell gets damaged and that hinders the DNA.
All right, the cell gets damaged and hinders the DNA, and that is what causes most of cancers. How can a cell get damaged? What kinds of things? And it's a lot of things in our environment.
Like carcinogen?
Yeah, what's a carcinogen?
Like a chemical dangerous to a cell would cause mutations.
Yeah. A chemical that we know is dangerous to the cell. Why is it dangerous to the cell? Is because it causes mutations which then ultimately can lead to cancer. And so carcinogens are things that we have known and have proven that exposure, cells exposed to this particular chemical induces mutations that will eventually lead to uncontrolled cell division in cancer. And so there's a whole host of chemicals that that's the case for. So like nicotine would be an example of that in cigarettes and so on. Yes?
Radiation.
All right, another thing is radiation. There's different kinds of radiation. So most common one that we're exposed to is ultraviolet radiation from the sun. And so radiation, absolutely, it causes the change in the DNA and can cause cancer. And so with UV radiation, it is affects our skin because that's what the rays, the radiation hit your skin and therefore induces mutations there and can cause skin cancer. Absolutely. There are other kinds of radiation like X-rays and so on. So there's different types of radiation.
I was just wondering, do you know how when you get sick or something, and then something else happens and there's complications? Could cancer be a complication? Like, could a cell's DNA be damaged from like a physical wound or something to have it like alter the DNA?
Not just a physical wound, but maybe something invading through there. So another example I was going to use about cancer-causing things and inducing mutations, viruses. Some viruses have been shown to cause cancer. You heard of the Hpv virus? It causes cervical cancer, and that's a known virus that induces mutations that causes cervical cancer, and so on. So I guess indirectly, it's not due to the damaged wound, but like something invading the body. Absolutely. All right, so that's the next bullet I want you to write here is what causes this transformation process. So, mutations in certain genes due to chemical carcinogens, which you guys talked about, X-rays or other high energy radiation, which we talked about, and some viruses. These are not the only causes, so like errors in DNA replication is certainly a cause as well. So there are other causes. These are just main, broad categories that cause a lot of different types of cancers. And so notice it says mutations in certain genes because a mutation in a gene that makes a protein that's going to get deposited in your skin to give you skin color necessarily won't cause cancer. All right, and so you won't get that protein that's going to be involved in making being deposited, like a pigment being deposited in your skin, but it won't cause cancer. So there's certain genes that mutations in them will cause cancer, and those are genes that have to do with the regulation of the cell cycle. So any genes that regulate the cell cycle, if the mutation happens in them, then you can result in uncontrolled cell division.
Are certain areas more susceptible to certain like reasons for the process?
Well, I guess, like ultraviolet radiation, skin would be an example of that. So like taking in nicotine, like your lungs, and tobacco chewing, you get mouth cancer a lot, and so on. So I guess they would be susceptible based upon what's causing the cancer and where it's at, in proximity. All right, so and then certain viruses, like I used the example of the Hpv virus, that virus attacks cervical cells, and so therefore, that's specific, to that. Okay. All right, so I want to talk about two classes of genes that if you get a mutation in can cause cancer. So I want you to be aware of these. The first class of the genes are called, it's a lot here, proto-oncogenes. This is going to make you appreciate my phone. These are called proto-oncogenes. These are normal genes that code for proteins that stimulate normal cell growth and division. So these are normal genes. So these proto-oncogenes are normal genes that produce proteins that stimulate the cell cycle. And so that's normal. So like when would that happen? Like when you cut yourself. So then the mitosis needs to occur to repair yourself. And so these proto-oncogenes are a class of genes. So it's not just one gene. There are many different genes that make in this class of genes that make proteins that stimulate cell division. But if those proto-oncogenes DNA is mutated, what can happen is that that it produces even more protein to stimulate even more cell division, and that's where you can get more uncontrolled cell division and loss of cell cycle control. All right, and so this is where we'll stop. Tomorrow I'll talk about tumor suppressor genes. All right.